Murder at the thermocycler. Note, any information discussed and the gel figures displayed in this video have been taken from the following source. If the information comes from a different source, it has been clearly indicated throughout the video.
It was a typical, mundane day in the biochemistry department at the University of British Columbia's Okanagan campus. Postdoc Sally Amino was working as usual on her research, minding her own business, and furthering her studies to hopefully one day teach a biochemistry class of her own. Unfortunately, she would never get that chance. Around 12 p.m. on January 27th, 2021, a horrific and piercing scream was heard by a colleague down the hallway. The authorities were immediately called, samples from the scene taken, and now it is up to our scientists and their DNA fingerprinting skills to determine who is at fault for the gruesome murder of the innocent victim. My name is Selena Spence, and I've been hired on as the primary investigator of this case. At the scene of the crime, there were drops of blood on the floor, and have been determined not the blood of the victim, so must be of the murderer. The murder weapon was determined to be this knife. Although this knife contains the blood of the victim, we have extracted some of the skin cells from the handle that will contain enough DNA to perform analyses.
So here is our poor victim, Dr. Sally Amino. Now, I know that she does have a shocking resemblance to me, but I can guarantee you that we are in no way related. Suspect 1. Dr. James Tography, a colleague of Dr. Amino. His potential motive being that he was upset that Dr. Amino recently beat him for a prestigious research grant. Suspect 2. Chromatography, the wife of Dr. Tography. Her potential motive being that she is angry that Dr. Amino beat her husband for that prestigious award. Suspect 3. Ferdinand polyacrylamide, a graduate student working in the lab alongside Dr. Amino. His potential motive? Perhaps he was angry that Dr. Amino was always signing up to use the PCR thermocycler when he needed it. And our final suspect? Suspect 4. Michaelis Burke, the boyfriend of Dr. Amino. His motive? Perhaps he was angry that Dr. Amino spent all of her time in the lab instead of with him.
All 4 of these suspects were seen by some students and faculty coming out of the lab at some point around the time of Dr. Amino's murder. Now let's get started into the details of the techniques I will use to rule out the murderer. For starters, let's briefly introduce DNA profiling. DNA profiling is when molecular genetic techniques are used to tell different individuals from others based on their DNA, whether that be distinguishing a human from another or distinguishing between different animals. DNA profiling has many different applications, such as identifying missing people, identifying bodies from disasters, paternity testing, determining species, etc. But of course, for this, we are using it for a crime scene investigation. DNA can be taken from all types of body tissues like blood, hair, saliva, semen, bones, soft tissue, and skin. And as already mentioned, I will be using the blood and skin samples of the murderer. Each of the suspects have provided their blood samples so I can compare them to the crime scene blood sample. Interestingly, 99% of human DNA is the same among different individuals. This 99% includes RNA coding regions like tRNAs or mRNAs that will encode for certain proteins, essentially those that have been conserved evolutionarily. That 1% that differs among individuals are referred to as polymorphic sequences. These sequences are the ones I will be looking at for the analyses. Specifically, I will be looking at anonymous sequences, that is, sequences that are not involved in any distinguishing traits and do not code for RNA or mRNA. It is important to use these anonymous sequences to maintain confidentiality of the suspects. We do not want to be looking at genes that could be involved in certain diseases or specific traits for privacy reasons. There are a few techniques I'll be using. These are RFLP analysis, BNTR analysis, and short tandem repeat analysis. The proper technique to use depends on how much DNA is available and the quality of that DNA. RFLP analysis can be used when there is a higher quantity of DNA. So I can use RFLP for the DNA taken from the blood on the floor of the crime scene.
RFLP stands for restriction fragment length polymorphism. This type of analysis uses restriction enzymes to cut at their recognition sites on the DNA.
Restriction enzymes are endonucleases that cut specific DNA sequences. They actually cut the sugar-based phosphate backbone of both of the strands. Digestion with these enzymes causes the DNA to be cut into fragments. This is possible because changes in the nucleotide bases in certain regions of the DNA between individuals will either make up the restriction endonuclease recognition sites or cause it to not be present.
For this analysis, I will be performing a double digest using the restriction endonucleases ECO-R1 and PST1. Here are the recognition sites for both ECO-R1 and PST1, with each of these red lines representing the cut sites. Any slight differences in the nucleotide sequence will make one of these sites not present. For example, if this A is swapped for a T, the site is no longer present because the ECO-R1 can only recognize that exact sequence. So for this example, this single strand has the cut sites for ECO-R1 twice, as shown in green. ECO-R1 will come along and cut here, and leave behind 3 fragments. But of course, this is just showing one strand, where in a real scenario, both strands are cut into fragments.
Different people will have a different number of these restriction enzyme recognition sites, and the sites will be located in different regions, resulting in different size fragments and a different number of fragments being produced after digestion with the restriction enzymes. To distinguish between the different size fragments, the DNA samples after digestion will be run on agarose gel, which will separate the fragments based on their sizes, with the shorter fragments traveling further down the gel and the larger fragments staying closer to the well where the DNA was originally inserted. The process creates an RFLP profile, or genetic fingerprint, which will be used to help identify the murder. A DNA pattern or fingerprint is referred to as a fingerprint because it is a unique pattern between each individual. Different people's DNA will produce different size bands and a different number of bands, so the investigator can tell who is who just from the DNA. The inheritance pattern of these restriction enzyme cut sites is codominant and Mendelian, so one can tell the difference between one allele inherited from the mother and one inherited from the father.
So, to visualize the fingerprints, the DNA is loaded into the wells here. A current is run through the gel, and this causes the DNA to run from the negative side to the positive side, as DNA is negatively charged. From this image, we know that this is a smaller fragment than this fragment because it was able to move through the gel further and faster than the other. This lane represents the profile or fingerprint for one person, and this lane is an example of the fingerprint of another person, showing that they are unique from one another.
So, if a pattern of suspect's DNA does not match the pattern of the crime scene DNA, then it can be determined that that suspect is not the murderer.
Here are the materials I will use to prepare the restriction enzyme digest. Firstly, I need to take the DNA that I sampled from the blood on the floor of the crime scene, distinguished as CS1. Then, I need to take the DNA samples from each of the suspects, suspect 1, suspect 2, suspect 3, and suspect 4. I will then take a restriction enzyme master mix, or ENZ, which has the 2 restriction enzymes and some buffer, and I will also need a DNA ladder, which includes DNA fragments of known sizes, as a guide, and use the ladder to determine the sizes of the sample fragments. To start off, the heating block is set to 37 degrees Celsius, as this is the incubation temperature of the digestion reactions. Then, 10 microliters of each DNA sample is placed in a microtube, and 10 microliters of the master mix is added to the tubes as well. I will flick each of the tubes to mix them and spin them in a centrifuge at 13,000 RPM for 1 minute to get the DNA and the restriction enzymes down to the bottom of the tube. After centrifugation, the tubes are placed into a heating block that is at 37 degrees Celsius for 45 minutes.
Now for the preparation of the egg roast gel. For visualization of all events, I will prepare a 1% egg roast gel. This takes 0.5 grams of egg roast and 50 ml of take buffer into a 250 ml flask. Now swirl the flask in a manner to prevent the formation of bubbles. Cover the flask and microwave it for 30 seconds on high. I now check to ensure that the egg roast is completely dissolved. If it looks good, then I can move on. If it wasn't completely dissolved, then I have to microwave it a bit longer. And now I need to let the solution cool at room temperature. Once cooled, it is time to add the cyber-safe fluorescent dye. I added 5 microliters of this dye to the solution, and this ensures that when the DNA is run on the gel, they'll be able to be seen. How it works is cyber-safe squeezes in between the DNA bases, or in other words, binds in between them. So when placed under UV light, the DNA fluoresces green. Without the dye, can't see the bands.
I will now pour the solution into the gel tray with the gel comb in place, ensuring that no bubbles are formed. This gel must sit so it can solidify for about 15 to 20 minutes. Once the gel is set, it will appear opaque, and I can now remove the comb from the gel, which leaves behind the wells, where I can eventually insert the DNA.
The gel tray is placed in the gel box, ensuring that the wells are closest to the black negative electrode, and this box is filled with more tape buffer, ensuring that the gel is covered by about 2 millimeters of buffer. After the 45 minutes of incubation, I now take the microtubes containing the digest out of the heating block and add 5 microliters of blowing dye to each tube. I put the tubes as well. The purpose of the blowing dye is to be able to track the sample's movement as it travels down the gel by determining the DNA movement rate. Another purpose is that the dye makes the DNA heavier or denser with glycerol, allowing the samples to sink to the bottom of the wells, otherwise the DNA would just float away. The tubes are then placed in the centrifuge, running at 13,000 RPM for 1 minute.
Back to the gel, 10 microliters of DNA ladder, which is the Hind 3 digest of phage lambda DNA, gets loaded into the first well. Each sample at 20 microliters is then loaded into the subsequent gels, as shown here. The lid is now placed on the gel box, and the box is connected to power to run a current through the gel. This arrow shows how the DNA will run down the gel.
I now run the gel at 100 volts. If the gel is running correctly, there will be bubbles in the tape buffer. After all the samples have left the wells, I crank up the voltage to 120 volts and run the gel for about 45 to 60 minutes. It is important to check the gel approximately every 10 to 15 minutes for the bubbles to make sure it is functioning properly. Also, it is important to make sure that the samples will not run off the gel, otherwise they'd be lost and further analysis could not be computed. Once the dye has traveled 2/3 along the way of the gel, this is when you can turn off power. The gel can now be taken out of the box and observed under a UV transilluminator in the UV gel dock system, and pictures of the gel can be taken for the RFLP analysis. Our RFLP profile is ready to go and our murder can be identified.
But before we get into those methods, let us go through an overview of a very important process in molecular genetics called PCR, or polymerase chain reaction. This is a useful process that allows for the amplification of small amounts of DNA into millions of copies. PCR reactions are similar to DNA replication as it produces many copies of DNA. The following are the necessary components in a PCR reaction. Template DNA, or the sample DNA, is required. Primers are short multinucleotide sequences that are designed to bind to specific regions of the template DNA and help facilitate the addition of new nucleotides to build a new strand. DNTPs are precursors to the DNA nucleotides that get added to a new growing chain to extend the primers, or in other words, create a new strand identical to the template. DNTPs complementary to the template strand are added by the enzyme DNA polymerase. A special type of DNA polymerase, Taq polymerase, is used in the reaction mix because it is adapted to function at high temperatures. It can live in temperatures up to 97 degrees Celsius, so it does not get denatured in the high temperatures that will separate the DNA. I will elaborate on that shortly. One more important ingredient is the buffer containing the magnesium chloride, which is required for the polymerization or growth of new strands to occur. All these components aside from the DNA template can come together as a master mix. PCR reactions are performed in a machine called a thermocycler, and each PCR cycle contains 3 steps: denaturation, annealing, and extension. In the denaturation step, the thermocycler goes up to a temperature of 94 to 96 degrees Celsius, and this causes a double strand of the DNA to separate or denature into 2 single strands. Since Taq polymerase is adapted to these high temperatures, its function is not disrupted by this heat as it can handle up to 97 degrees Celsius. In the annealing step, the temperature is decreased between 45 to 65 degrees Celsius, and this temperature allows for the primers to attach or anneal to the single-stranded DNA by hybridization. They are complementary to the regions on the DNA, and this represents the starting point for the DNA synthesis. And finally, there is the extension step, where the temperature is increased to 72 degrees Celsius, and this allows for Taq polymerase to add nucleotides for the DNTPs that are complementary to the template strand to extend the primer and synthesize a new strand. This is a simple representation of the first cycle of PCR. We can see the 2 strands being denatured. The purple bars represent the primers annealing, and the blue bars represent the new strands that have been made by Taq polymerase adding on DNTPs. The same cycle will repeat about 25 to 35 times. The exact number of cycles just depends on what will cause the highest yield. Note that during the first cycle of PCR, the denaturation step occurs for a longer time, about 2 to 4 minutes, to make sure that there is complete denaturation or separation of 2 strands. In addition, in the final cycle, the extension step is carried out longer for about 4 to 10 minutes, depending on if doing an STR or a VNTR. Now that you have a good understanding of PCR, we will dive into the STR and VNTR techniques. Firstly, let's talk about STR. If your DNA sample is very small and is partially degraded or not in the best condition, STR is the best method to use. STR stands for short tandem repeats. In this investigation, I will use STR analysis for the skin samples that I have taken from the murder weapon, as the DNA here is not the best quality and there's not a lot of DNA here. STRs are short repeats that are usually about 2 to 9 base pairs in length and are present in both anonymous and non-anonymous regions. When there are small amounts of DNA, PCR analysis is required to obtain many copies of the DNA. Since STRs are quite small, PCR amplification can be carried out effectively, and partially degraded DNA can be used to get many copies for analysis. Think of it this way: if the DNA's sample is very small, but the repeats are small, there's a greater chance that the pieces of DNA that are still intact will include the short repeats than, say, a repeat that was 100 base pairs long. The samples don't even have to be fresh. If the samples are properly stored, old samples like blood and buccal cells can be examined by STR profiles, and these profiles can be of good quality. Today, we'll be looking at the STR locus that is used for DNA profiling, called the TH01 locus. There are several different alleles of this locus. This locus is made of the sequence TCAT, and each individual will contain a different number of these repeats, allowing for the differentiation between the DNA of different people. Not only are there different number of repeats between individuals, but there are also differences between 2 chromosomes of the same individual, making them heterozygous. For this analysis, I will be using a simulation TH01 allele ladder. This ladder does not show the true molecular size or the number of base pairs of the fragments. Instead, it shows the number of repeats that are present in each band, so each band represents a different allele for that locus. Since the different alleles are different sizes, we can use gel electrophoresis to differentiate between them. Now, there is this specific software program called CODIS, which stands for Combined DNA Index System.
And this is used to make a database of DNA collected from crime scenes and suspects and the criminals themselves. It can be used for investigations of serial crimes, unsolved cases of offenders that have previously been entered in this database. It can also be used to identify missing people. It is used in the US, Canada, and numerous other countries, but what is special about this is it uses 13 different STR loci, whereas here we're just looking at one. And these loci are present throughout the entire genome. Additionally, a Y chromosome STR can be performed to help identify perhaps a male who sexually assaulted a female, as only males possess the Y chromosome. Many kits have been made over the years, and the discrimination between different individuals continues to get better. For instance, now results can be determined in under 5 hours, and the sample could have come from a very tiny little speck of blood on the floor.
As mentioned, CODIS uses 13 STR loci for profiling, and additionally, sometimes a Y chromosome STR. Using multiple loci allows for simpler and better differentiation between individuals' DNA profiles. If only one locus was evaluated, there is a much higher chance of the number of repeats being identical, thus making it harder to distinguish between individuals as their profiles could look the same. With more loci, the probability of two profiles being exactly the same by chance decreases, and it becomes nearly impossible for two profiles of different individuals to be the same. Also, the myelogenin locus is commonly used as it allows investigators to determine an individual's sex because the gene is present on the human sex chromosomes.
So the X and Y chromosomes. The PCR products are different sizes depending on whether it is an X chromosome or from a Y chromosome. Thus, the more loci that are studied, the better the confirmation that the profile that matches the crime scene profile is indeed the murderer and not just the same by random chance. It is important to note that the loci chosen are all anonymous sequences so that confidentiality is maintained and so that no medical information is seen. However, there is still some controversy surrounding the storage of people's DNA sequences as people fear it will be used incorrectly, and they fear it's not fair to have this private information of people that are later determined to be innocent.
And now onto VNTR. VNTR stands for variable number of tandem repeats. In the genome, there are multiple different regions containing VNTRs, and VNTRs are still short sequences, but much longer than STRs, as they can be between 10 to 100 base pairs in length. Like STRs, the number of repeats between individuals will differ. Using restriction endonucleases, the extra DNA on either side of the repeats can be cut off, leaving behind just the VNTR region. The number of repeats can be compared amongst different people. Like STR analysis, PCR amplification can be used to get many copies of the VNTR region. Note, however, that the DNA needs to be in good quality to use VNTR, unlike STR analysis. If the VNTR region has been disrupted, the analysis will likely be inaccurate.
For both STR and VNTR loci, the DNA sequences that extend on either side or flank those loci are usually identical. This makes primer design for these regions really easy because the same primers can be used. It makes these flanking regions easy templates to which the primers can anneal, so PCR is nice and easy. Now let's just back up and discuss the mode of inheritance of these repeats. The number of repeats, whether STR or VNTR, are inherited in the way of Mendelian genetics, but they are codominant. That is, one allele inherited from the mother and one inherited from the father. For diploids like us, the number of repeats on a chromosome that we got from our mother may be different from the amount of repeats from the chromosome we got from our father. The locus is the region on a chromosome where the repeats of a particular sequence are located. The number of repeats on the chromosome is considered an allele.
Here's an example looking at the TH01 STR repeat locus. Say your mother has 4 of these repeats on both of her chromosomes, or in other words, contains the allele with 4 repeats, and your father has 5 of these repeats on both of his chromosomes, or contains the allele with 5 repeats, as shown here. But since both of these alleles have the same number of repeats on their chromosomes, these individuals are considered homozygous at this TH01 STR locus, the genotype of the mother being 4,4 and the genotype of the father being 5,5. And yes, that makes them homozygous. Now you, their offspring, will get 1 copy from your mother and 1 copy from your father, giving you 1 chromosome with 4 repeats and 1 with 5. Since you have a different number of repeats, this makes you heterozygous at this locus. The genotype is 4,5. So they're different, so you are a heterozygous.
So for this investigation, I will look at the different number of repeats found at the crime scene and compare that to my cheek cell DNA in VNTR analysis to rule myself out as a contaminant of the crime scene DNA, and STR analysis will be used to compare the crime scene DNA to the suspect DNA to determine the culprit. But before we get into the protocols, I want to briefly discuss the pros and cons of each the RFLP, STR, and VNTR analyses.
A benefit of RFLP analysis is that it is easy to distinguish between different samples based on the bands on the gel, especially if one sample has more bands than another. Also, the extra time it takes to perform PCR is saved as there's already a large sample of DNA, so no amplification is required. However, the limitations to this technique include that there needs to be a high quantity of DNA in a sample to perform this. This DNA needs to be of good quality or not degraded, and it is often difficult to determine when bands are identical or coming from the same person. Also, the proper restriction enzymes must be chosen and the recognition sites must be present in the DNA strands. STR analysis. Some of the benefits of STR analysis include that small amounts of DNA and lower quantity DNA can be used. The identical flanking regions on either side of the repeated regions allow for easy primer design, and the PCR amplification allows for millions of copies of the target DNA to be produced. Also, these sequences are highly polymorphic, making them variable amongst individuals, and there are many commercial kits that can be purchased for these analyses to minimize the amount of preparation required initially. As for a limitation to this technique, if someone is only analyzing one STR locus, there are increased chances that different people will have the same number of repeats and their bands will look identical. Therefore, it is important that many different STR loci are examined, as this decreases the likelihood of different individuals having exactly the same numbers of repeats at all the loci analyzed. The benefits of the VNTR are the same as those of the STR analysis, except that with the VNTR, the DNA needs to be of higher quality, in other words, cannot be degraded.
I will start by explaining the VNTR protocol. Here's a quick rundown of this process before I get into the details. As an investigator, there's a possibility that my DNA could have contaminated the crime scene DNA, so in order to ensure that this is not the case, I need to test my DNA against the crime scene DNA. I'll do this by extracting DNA from some of my cheek cells. I will then use PCR to amplify the D1S80 VNTR locus, and then I will run the PCR product on a gel to visualize the bands. Then I will take some of the crime scene DNA from the skin cells in the knife, amplify the same locus, the D1S80 locus by PCR, and run this on a separate gel.
The D1S80 locus is located in an anonymous region and is on the 1p chromosome's distal end. Of course, the alleles for how many repeats are present are inherited codominantly and is Mendelian. Each repeat contains 16 base pairs, and there are usually between 14 to 40 repeats of these 16 base pairs present. Most commonly, people are heterozygous for these alleles, as it is extremely rare for people to have exactly the same number of repeats.
Firstly, I need to isolate some of my own cheek cells. To do this, I need to chew on my cheek carefully, to loosen up some of the cells, and rinse and swish with about 10 to 15 ml saline solution for 1 minute. After the 1 minute, I will spit this into a cup. Yummy. Next, I swirl the cup, resuspending the cells, and transfer 1.5 ml of this into a 1.5 ml screw cap microcentrifuge tube. Note the importance of the screw cap. This prevents the cap from flying off and losing some of the sample when the samples get boiled later on.
Now put the tube in a centrifuge for 2 minutes along with the balance at 13,000 RPM. Note, There must be a clear supernatant after the centrifugation and a white pellet at the bottom that is about the size of 7 to 8 millimeters. Centrifugation must be carried out again for 30 to 60 seconds if the supernatant is still cloudy.
I will now dunk the supernatant into bleach. Carefully not to dump out the pellet with it because the pellet is where the cells are located. With the rest of the 1.5 mils of the cell suspension, I will repeat the same steps again with the pellet still in the same centrifuge tube so that the pellet increases in size. Now that I have this large pellet, I resuspend it in 140 microliters of lysis buffer and mix it by pipetting or vortexing.
The following shown here are the components of the lysis buffer. The chelating agent gets rid of the magnesium that is required for enzymes that degrade DNA and for DNA polymerase. The proteinase K digests proteins that are in the solution but does not affect the DNA.
The tube will now be incubated in a water bath for 15 minutes at 55 degrees Celsius. After the 15 minutes are up, the sample is vortexed for a duration of 20 seconds.
I will now place the tube in a heating block set to 100 degrees Celsius for 15 minutes, and then let it cool at room temperature for 2 minutes. I then vortex the tube for 10 seconds to ensure the solution is all mixed up, and then centrifuge it at 6,000 RPM for 2 minutes to create a pellet. The DNA is in the supernatant.
Be careful that you do not discard the DNA. I'm not going to make that mistake twice. Anyways, so now I'm going to take 80 microliters of the supernatant and transfer it into a clean microcentrifuge tube. It is extremely important that none of the pellet gets transferred with the supernatant. Otherwise, the Chelix beads can trap the magnesium, preventing it from being used by Taq polymerase, and this results in zero PCR products. And now, I put this tube on ice for 3 minutes.
Now here we have 2 PCR tubes that both contain this EDBO bead, and one of them gets labeled SCS2 for the crime scene DNA and has been isolated from the skin cells on the knife, and the other SCH, which will contain the DNA that has been isolated from my cheek cells. Then I will add 20 microliters of the D1S80 primer mix to each of these tubes, and then 5 microliters of the crime scene DNA to the one tube, and 5 microliters of the cheek DNA to the other. I now invert the tube to ensure that all the EDBO bead gets dissolved.
Tubes are now placed in microcentrifuge tubes that don't contain caps, and this is so that the tubes can fit in a centrifuge and spun for 1 minute at 13,000 RPM to get a pellet at the bottom of the tube.
And finally, PCR tubes are put into the thermocycler, and the PCR reaction is performed as previously described. After 3 hours, we now have millions of copies of each.
In order to visualize the bands, I need to prepare another agarose gel and perform an agarose gel electrophoresis. I will take the PCR tubes, put them in the microcentrifuge tubes so that they fit in the centrifuge them for 1 minute at 13,000 RPM. I now add 5 microliters of blue loading dye to my 2 samples and mix them by pipetting up and down. Once again, I centrifuge them for 1 minute at 13,000 RPM to pull everything down to the bottom of the tube. I also prepare the gel as previously described, except this time I will be using 3% agarose instead of 1%, and this means that I weigh out 1.5 grams of agarose into a 250 ml flask, 50 ml of tape buffer, and let it cool for 10 minutes because this gel solidifies a lot faster than the 1%.
I load in 20 microliters of my PCR amplified VNTR cheek cell DNA, and in the next lane to this, I load in 10 microliters of the EDBO-Quick DNA ladder. All the bands in the ladder are of known sizes, so using this ladder I can generate a standard curve and determine the size of the bands from the samples from there. The other wells will be filled with the STR samples, which will make sense in a bit. Then, I take another 3% agarose gel that I've previously prepared and add 10 microliters of the EDBO-Quick DNA ladder into the first well, and then 20 microliters of my PCR amplified VNTR crime scene DNA to the second well. These gels will now be run at 100 volts until the samples have all left the wells, and then the voltage will be increased to 120 volts. Once the dye front has traveled 2/3 of the gel, I stop the voltage, remove the gel from the gel box. Just like described before, I put the gels under UV transilluminator, take pictures of the gels. Don't worry, I'll show you all the gels once I get to the data analysis part. And now, the STR analysis procedure to determine the killer. I have extracted DNA from the skin cells from the knife. 10 microliters of each DNA sample is placed in the microtube, and 10 microliters of the master mix is added to the tube. They're put in a nice bucket, too. I now take 20 microliters of the PCR master mix that includes all the necessary components that I discussed earlier, and this was stored on ice, and add that to each of the tubes. Then, I add all the DNA samples to their label tube, flip the tubes to make sure everything is mixed, and centrifuge them at 13,000 RPM for 1 minute. Then, I put them in the thermocycler to perform the PCR reaction. I prepare 3% agarose gel, just like for the VNTR. I take the PCR tubes and I centrifuge them inside of the microcentrifuge tubes, of course, for 1 minute at 13,000 RPM. I am sure you're starting to see the pattern here. And then, I add 10 microliters of orange G loading dye to all the tubes, and pipette them to mix it all in, ensuring that a new pipette tip is used for each tube. You do not want to contaminate your samples. That would be very bad. And then pulse spin them all again. Now I'm going to load all of these into the gel that the VNTR products are already in. 20 microliters of the TH01 allele ladder to the first well, and then my 5 STR-PCR samples into the following wells. I then perform the gel electrophoresis, as I already explained, turn off the power, unplug the electrodes, take the gel tray out of the electrophoresis box, and observe my beautiful gels under UV light. And now for the exciting part, the DNA analysis. First, I will start with the VNTR analysis, because I do not want to reveal who the murderer is too soon. Here is the gel from the VNTR analysis. It shows in lane 7, the cheek cell DNA band. In lane 8, the EDBO-Quick ladder. In lane 9, another EDBO-Quick ladder, as this image actually shows 2 gels that were put together. And in lane 10, the crime scene DNA from the skin cells. Since there are 2 different gels used, there need to be 2 different standard curves created in order to determine the band sizes of the samples. And those there are the band sizes of the ladder. This table indicates the distances that the DNA fragments are traveling down the gel in millimeters and their corresponding size in base pairs. With a ruler, I have measured the length from the end of the well to the middle of each band for both ladders, the cheek cell DNA and the crime scene DNA, as indicated in green on the screen. The DNA ladder fragments are already of known sizes, but the sizes of the sample bands are still unknown, and they're shown there in red. I have generated some standard curves. Here I have taken the distance that the bands traveled from the DNA ladder corresponding to the gel containing the cheek cell VNTR PCR products on the x-axis and plotted against the known band size on the y-axis. The y-axis was log transformed, and the exponential trend line was added. Note that I have omitted one of the bands from the trend line, largest band, to make for a nicer trend line with an r-squared value that's closer to 1. I have done the same procedure as with the previous graph, except I'm using the DNA ladder corresponding to the gel containing the crime scene VNTR PCR product. These 2 graphs, I have taken these 2 equations. I can now plug in the values of the bands from the samples that I measured with the ruler, and this will give me the approximate sizes of the bands in base pairs. Here is an example calculation for the cheek cell VNTR product. I plug in the distance traveled in millimeters, so 28 millimeters for x in my equation. This gives me y equals 587.1, so this fragment is 587.1 base pairs in length. Here is the completed table with the unknowns filled in using those equations to find those values. What I've done is I've taken the sizes of the bands and related them to a table that will give me the approximate number of repeats present in the bands. Here's just a portion of the table. For one of the bands of the crime scene DNA that was 417.8 base pairs long, I picked the allele size that is the closest to it, and from there, this tells me the number of repeats that are present, and I put these values in my table. You will notice that I have 2 bands for the crime scene DNA and only 1 band for the cheek cell DNA. This indicates that the murderer was a heterozygote, and I am a homozygote for this BNTR region. The genotype is denoted as the number of alleles that are in each chromosome. So for this heterozygote, those 2 numbers are different, and for my DNA, the number of alleles in both chromosomes are the same. This is why we see 2 bands for the heterozygotes, because the bands are different lengths, and only 1 band for homozygotes, because both chromosomes have the same number of alleles and are therefore the same size and travel the same distance.
So what is the verdict of the VNTR analysis?
My DNA did not contaminate the crime scene DNA, but now on to the more exciting stuff. Folks, we are getting closer and closer to finding our murderer. Here is the gel from our large sample of DNA from the blood on the floor. The sizes of the bands in the DNA ladder are indicated to the left of the gel. I carried out the same procedure as I did for the VNTR analysis. So quick summary, I measured with a ruler the distance that the bands travel, in green, generated a standard curve with the distance migrated in milliliters on the x-axis, and the band size in base pairs on the y-axis. Once again, I removed the largest band size from the data to make the trend line a lot smoother. Now that I have a graph, I can get rid of all these question marks using this equation. And the calculation was done the same way as previously demonstrated. In green are all the sizes in base pairs of all the sample bands that I've calculated using that previous equation. So let's compare, shall we? Looking at the largest fragments from each sample, it appears that the closest suspect value to that of our crime scene is our third suspect. Onto the second largest band, it appears that once again, the fragment size of suspect 3 is closest to the crime scene. And finally, onto the smallest band, the trend is the same. When we look back at the gel, it also looks like the bands of suspect 3 look the most like the bands of the crime scene. So, does this mean that suspect 3 is the murderer? Well, it looks promising, but I'm going to check out our STR results and see what they reveal. Here is the STR gel. Circled here are the number of alleles that are present in DNA fragments in each band. So the top band has 10 alleles, the bottom band has 3. This is how we will determine each person's genotype for the TH01 STR locus. For the crime scene DNA, it looks like the genotype is 8, 5. For suspect 1, it looks like the genotype is 9, 5. For S2, it looks like the genotype is 7, 4. For S3, it looks like the genotype is 8, 5. And for S4, it looks like the genotype is 9, 4. I have compiled all the genotypes into a table for comparison. And what do we have here? It looks like the match in genotypes is once again between the crime scene DNA and suspect 3.
Alright folks, it is the moment you have all been waiting for.
Here are all the suspects. It was not Dr. Photography, not his wife Roma, and it looks like the boyfriend is off the hook too, as our results clearly indicate that the master student Ferdinand polyacrylamide is guilty.
I've spoken to the 3 suspects that have now been cleared. I simply asked them why they were present in the lab that day.
That is where I work. Of course I would be there on a weekday. I cannot believe that I was accused of doing something like that to my poor colleague.
I just thought I would surprise my husband with a nice lunch. Boy, that Sally was a nice girl. It saddens me to hear the news.
Michaelis Burke, Sally's boyfriend, was too distraught to be featured on video, but he remarked that he was visiting Sally to drop off her phone that she had forgotten at home that morning. Now, it definitely seems like an odd motive that Mr. Polyacrylamide had. Sally was reserving the time with a thermocycler, fair and square. I would like to think that having him behind bars is not only good for Sally's sake and the safety of others, but also for the sake of biochemistry. I mean, come on. A master's student who doesn't know that he was leaving his DNA all over the murder weapon and crime scene? Yikes. Well, this concludes my investigation. I have used VNTR analysis to make sure that my own DNA did not contaminate the crime scene, and RFLP and STR analyses to catch the murderer. I hope you have learned lots about the importance of biochemistry and DNA analysis in crime scene investigations, and how important it is to make sure you don't leave your DNA behind if you're going to try and commit a terrible crime. We will catch you.
Excuse me. Are you Ferdinand polyacrylamide?
Yes, that is me.
You are under arrest for the murder
no,
of
no,
Dr.
no.
Sally